to one and all present here. We are in the 5th day. From tomorrow we will be missing you all. Let us start the today's session. I welcome Dr. Nagaraj Nagatihalli, Assistant Director, Basic Research Center and Associate Professor of Surgery, Sylvester Pancreatic Cancer Research Institute, University of Miami, Florida, USA. And I welcome Dr. Chandrasekhar G. Joshi, Coordinator of MSc Biochemistry Program and Coordinator of GAN Program. And I welcome all our participants and technical team. Have a great day. Thank you.
So good morning, everyone. So the last day, I think we're going to miss you all, as she said, today onwards. I hope you all enjoyed it, and you have to take the test, and hope you all pass. Today, I think yesterday's talk was we screen natural compounds. And then we select one of the natural compound, which is active compound, and then we study, right? And you saw, if you don't know what natural compound, if you don't know, what signaling pathway, what activates and all those, you kind of learn those. Also, once you know which the active compound, you know how to study. Okay, you can easily. Doesn't matter where you go back, and actually you can think of the same way, methodologies, they're all same. So you just start with it. Today, say for example, I have a specific target. Okay, I don't know the compound. I don't know the inhibitor. Okay, but I know the target. So what do? How do we do the research? How do we find the compound that can target my specific target that I find, right? So that is the story. I take CREB, cyclic AMP response element binding factor. So from day one, I'm talking about day one, day two. I think we talked about a little bit here and there. It's easy because I can tell you exactly the. Say for example, it may not be CREB, it may be AKT, it may be ERK, it may be other signaling pathways, it may be some other factor in some other cell type. It's okay, okay. But you know the target. Now it's my target is a CREB, okay. Now I want to find a drug or the chemical which actually inhibit the CREB signaling. That is the idea, right? So first of all, you need to know why you select the specific target, right? For example, I selected the CREB based on many factors I have investigated. I've done a lot of other research, and then I also published, and I know that this is important signaling pathway, right? That's how I selected the CREB. And also I know what activates CREB. Okay, the same way I told you many things about cell-to-cell communication, and you need to know what activates your target, right? The same way, same philosophy, right? So now we understood CREB is important, and we know all the targets CREB actually, can be targeted and inhibited or activated or something. So now we knew that CREB is important. It's an important transcription factor, actually that regulates many genes involved in cell survival, proliferation, metastasis. It's actually CREB in many cancer, just not pancreatic cancer that I showed. It's also expressed. I also showed you cholangiocarcinoma, gastric cancer, esophageal cancer, And many other cancer, Krebs also get activated in tumor when compared to a normal, right? Making it a promising therapeutic target. That's why it's overall, if you can target Krebs with a compound, it can actually study that same thing, same in other diseases also, where Krebs is getting activated. So that is the idea behind it. So then we thought of doing the compound screening, even though at that time when I found the Krebs, there were not many targets, I mean compounds that actually inhibit Krebs. There were very few inhibitors available in the market, even now. But then I thought of doing our own screening and find targets, either kinases, or just the natural compound, both actually. We were interested in anything that can hit the Krebs, right? We know now Krebs is one of the therapeutic target, and we understood the role of Krebs in cancer, just not pancreatic cancer. We studied all those things, we published also. But only thing what we did not publish is that drug therapy. Means we didn't have the drug at that time to inhibit the Krebs. So we can do knockout, we can do knockdown, we can do siRNA and all those things during that time. I'm talking about 15 years ago, right? And overactivation of Krebs also we kind of screened all the TCGA. TCGA actually came out long ago. I think I showed you one of the figure that database was there for long. So we were able to see the differences of Krebs expression in different cancers also at that time. So, and also we kind of rationalized why we want to target Krebs, right? So we kind of understood the pro-tumorigenic role of Krebs, and also during after the progression, metastatic. You saw all the data where we overexpress Krebs and actually it metastasized, the cells metastasize strong, faster. But only thing is I did not go through the molecular aspects of those studies, because it's again another avenue. But we were interested in the preclinical development and clinical development. So also I showed you we knew preclinical studies also, like for example, it indicated us that Krebs inhibitor is important, right? Then we started thinking about the compound libraries, right? So, synthetic compounds, I was just started thinking like, synthetic compounds, can we extract from natural product during those days I'm talking about. And also we were looking into FDA approved, drug library. There are libraries available where FDA approved libraries, even in India you can actually buy it. There are libraries available. You can actually screen those libraries. Like it's all in the 94 or the 384 well plates, 364 well plates. And also you have to think about the selection criteria. It's okay, for example, if this FDA approved drug library, if this is a natural compound library, natural product library, if it is a chemical library, how do we do the selection? How do we predict from this screening, these are the compounds are important, right? We had to do the cell permeability assay and pharmacokinetic profiling, all those things. But you already know, yesterday we talked about all these things. Then you have to design assay. For example, how do you screen? First of all, you know the target, but how do you start screening? How do you take all these compounds and like Krebs inhibitors, how do we do it? So for that, you have to think about first of all, you have to understand how do you measure the Krebs DNA binding, or you have to use some fluorescent binder, or you can do cell-based assay, but you can actually do reporter assay. So reporter assay is very easy. At that time, we were thinking about the reporter assay because we can actually, there is a luciferase tagged, Krebs-driven luciferase tag and GFP reporter systems available. So you can actually tag them and it actually gives you a color. And when the drug hits, then your color changes, things like that. So you can actually measure those color, fluorescence, things like that. So we designed the cell-based reporter assay for the Krebs. And then we kind of optimize those, whatever the luciferase tagged cells for the sensitivity, specificity, and also robustness, using the known CREB inhibitor. We already knew there were some few inhibitors available, at that time they were available. You had to compare though. So for example, if you don't know any inhibitor for the target, it's also fine. You can actually screen it. But for us, we had it. We had one or two inhibitors available. We kind of compared that. If you don't know, if, okay, for example, if you don't have the inhibitor, you can actually knock out the specific the target, and you can actually study it too and see, make sure that actually is working for you. Is either way, okay? You can actually do it. And then we have to set the activity threshold. Okay, if the activity, the CREB activity in the luciferase activity assay, I know how much activity is actually is good enough. You know, this kind of a threshold, you set it up. If you don't know, let me tell you, if you don't know, if you know the chemistry lab who actually knows the activity assays, they regularly they do those, and you can actually talk to them and discuss with them. Okay, that's what we also did. Okay, in the initial stages, and also you have to know the criteria for hit selection. You have to know the IC50 values, efficacy, selectivity of our CREB inhibition, and also initial dose response curves to determine the activity. You have to know all those things. You have to do all those small assays and determine, make sure that your activity or whatever that you want to do, conduct experiment. Because I'm giving you a hint that there is a validation of even before you do the compound screening, and for your target, be ready so that your target is like all the validation are ready, and now you want to, okay, if the validation is ready, I want to go in and do the compound screening. And also there are some secondary assays that you need to think about. Okay, like using the alternative assay formats, like for example biochemical assay, cell-based assay to validate the CREB inhibition. So you can do a lot of these things like groundwork before you screen. Also you can do the counter screen to assess the specificity against other transcription factors. For example, CREB you know already, so take another one and make sure that is not getting hit by the when you knock out CREB, and it should not affect the AKT. It should not affect some other ERK signaling or ERK factor. So you have to make sure that it is only hitting the CREB. That's what I'm trying, counter screen and make sure. Okay, that is hitting the target that you wanted. Okay, the validation techniques are dose response studies and all those things. Overexpression knockout, I actually discussed that before. Now we examined whether additional kinases like for example MEK-ERK and AKT also activate CREB. We knew in the KRAS signaling pathway, so are also involved in inhibiting phosphorylation. So what we did initial screening, we inhibited MEK-ERK and we also inhibited AKT, and we checked whether the CREB is getting inhibited or not. This kind of assays you have to do. Okay, for example, if you know that upstream factors of whatever the target that you're thinking about, so you better inhibit those and see whether they are the real upstreams or not. So it's just a validation, again, making sure your discovery of whatever the target is actually discovery was right. Okay, that means that you understand the biology of it before even discovering something compound and going forward. Because once you know the target, once you know the compound, you get excited and keep doing the experiment. You will forget about what exactly, how they're exactly getting activated also. So we also selected in the initial screening 2 pancreatic cancer cell lines. You can select like 2 or 3 multiple based on, if you want to study the resistance and the sensitivity, you can actually select resistant to and the sensitivity to and check all those. It all depends on what type of experimental parameter that you're following and want to do. Okay, we screened at that time, long ago. Okay, we screened like 80 kinase inhibitors. They were available, and also we had like 35 different kinases from the TalkRisk Screen kinase inhibitor toolbox. We actually available. This company TalkRisk Screen, actually you have a multiple FDA approved drugs, and also they have natural compounds also. They can they sell with a plate So we selected at that time. Now we don't have to go with these companies and all. We have NCI because I'm an NCI investigator, so we can actually get it from the NIH, everything. At that time I was not, so I was actually screening, making sure we get all these things long ago. Okay. Then you plan your high throughput screening. What do? You take all these cell lines, like you actually do all these 384 well plates. You put the cells on the plate, and you do the ELISA and you grow them, and then add the drug. And the drug means that here, when you get the drug, like there is a 94 well plate, you can actually take from the multiple pipette, and then you screen. This is again another technology. Believe me, it is. You can go wrong also. You have to know how to pipette, first of all, in this scientific research. I think Joshi was talking about all those small things that you need to know. I'm not covering those things. What I'm trying to tell you is that, so basically you take the drug. These drugs will come in 384 or 96 well plate. You can actually select based on that, and you can screen. And also there are now in our core facilities where they do it for us, okay, now. And means that the machine will do all those things. It's a computerized machine. Actually, they can do the drug screening for us, okay. They can do everything. And then you, because this is a luciferase tagged screening, so basically you can get the target kinases. Some kinases will get activated, some kinases get inhibited. Okay, you have to think what is actually your research is. Are you activating it, or or you're inhibiting it? You can select based on that. All this high or low expression of Krebs, and you can select those libraries and start thinking about it. You have to do research again and start searching for literature and see what they do. What we did is we selected one cell line, we screened, and we got these numbers, right? When there is a high number, there is a low number, there are non-selected one, there is one activated ones, these are all blue in the activated one. We actually have a total Krebs expression. We compared that with the phosphorylation. The same thing you do too in a smaller quantity. We actually did the kinase inhibitors. These are all the kinase inhibitors we selected, and we have these kinases here. So these are inhibitors, and these are kinases. So we can actually inhibit using all these inhibitors, and but your cells, your Krebs luciferase tagged cells are in the plate. So that means that whatever you hit, it is actually you're hitting the Krebs. Okay, so either the activity will go high or the low. So you have to measure those. So what we did, we measured the peak. We measure all the peak, and then we select from this. We normalize those, and we select the best out of it. So then we get that inhibitors of this phosphocreb. There are many, like many compounds will inhibit all these things, and then there are some activators. Some of them will activate also your Krebs. Okay, some will inhibit, some will activate. Okay, now it all based on your research, how you think, what you think about this signaling pathway, and how they are actually, these signalings are related to each other, and whatever that you think, you have to do the research again. Okay, you get great data, but what do you do with the data? So that's again research. So you have to think about it, and you have to have a plan and discuss with others and those who actually screen like this, and do it. Okay, and then one cell line is over, and we select the second cell line, and we make sure that we get the same hit or a different one, and then we start thinking about why different, why same. These questions will come. What mutation these cells have, and what is the proliferation rate, and how these cells behave. Very important. Okay, understanding all this. So then we get this hit, then we select, take these kinase inhibitors, and then hit it, and then we got this peaks, and the same like the other cell line, and we select the inhibitors for the phosphocreb and also activators for the Krebs. We select all those things. Now we combine all of them. Okay, and we get new targets. There are many of them. Okay, and we know these signaling pathways. These are known for Krebs and well known. I can tell you each one and what they do on the Krebs. And these are unknown. These are not knowing at that time that what these unknown signaling pathways do on Krebs. And these are completely unknown pathway that activate Krebs. Okay, because CDK Aurora, there was no literature available at that time on these ones. Okay, so you select based on this. Now, the next questions will come to you is that, okay, what is the difference between these two cells? And we got this common signaling, because what I'm saying is that there are common, pathways that you selected from these two cell lines. Like that, for example, there is a sensitive cell lines and resistant cell line. You also select based on the common, these cell lines. Just one cell line for the sensitive or the resistant is not enough. You have to make more cells, okay, different cell types, and also you screen all of them and select the kinases and the targets, and then think about it. What's going on? Remember, it actually, what you're not seeing is that they are inhibiting CREB. These are signaling pathways are activating CREB or the inhibiting CREB. In this case, they are inhibiting. Means that when you use the SRC inhibitor is also inhibiting CREB. That's what I mean to say. Okay? When you're taking the Aurora inhibitor, the inhibitor of Aurora is inhibiting CREB. Okay? Means that if you don't have CREB inhibitor, you can actually use CDK inhibitor that also should inhibit CREB. So then you go back to lab and start using the CDK inhibitor, which is FDA approved. Those compounds that we screened, all FDA approved compounds, and CDK inhibitor, one of the FDA approved compounds that we used, and now we can use that compound to inhibit our CREB. That means that if you don't have a CREB inhibitor, you can use the CDK inhibitor and do your experiments and conduct your study, but it still is working. But it may not be specific to a CREB, but CDK is also get activated during the progression of the cancer, so you can use it. Okay, CDK also get activated, and EGFR get activated, SRC, TJ beta, ROC, EEC, we are all getting activated. We talked about some of these during all these 4 days. There are these activators or targets that keep coming back because they're all common signaling pathways in pancreatic cancer. That's why they keep coming back. Probably if you take different cell type like breast cancer, lung cancer, probably they are different. You actually don't see them. These ones are coming back up again and again, okay? Okay, now the story is that there comes Joshi. So what he did, we had this screening, we were all getting ready, all the screening came. Then he said, oh yeah, I got the ICMR fellowship, I want to come and do, I gave a letter, and he got this fellowship and he wanted to come. So then we have to fix the time during when you should come, and when you should be able to start some of this work. And based on those things, I think when he was here, I think he came all the way to here. I think there was a lot of water, and then it's Miami is beautiful. It's like a mini Goa. In America, Miami is like for you in India, Goa. Miami is for US is Goa. So like that, it's fantastic place and the beautiful cancer center. And he came there, and it's overwhelming. Okay, it's entirely different. And it takes time. I mean, Satish was there, and the doing postdoc there, and it's an entirely different thing. So it's a great opportunity for a faculty from Indian faculty coming to US and learning. It's not US, any other country. You go to developed countries, there is different approach they give. That different approach they give to science. That's one of the reasons I was also passionate to go to countries like that and to learn. Okay. What he did, okay, he selected one of the compound that's called berberin. Okay, we had this natural compound screening to target CREB, and we had multiple of them, and then he wanted to do the berberin. So, and also he selected that berberin, and then very interesting that why he selected berberin, right? Berberin at that time there was not much literature available. Okay, this is again 2018, and also there was NCI China collaborative studies in 2017. He came in 2018. In 17 there was big literature, big program was going on between US and China where they were interested in the Chinese medicine, okay, on cancer research. And they were working on those, and the NIH was interested, and they were like, there is a cancer prevention laboratory, and also at the NCI, they studied the effects of berberin and other compounds extracted from these herbs on colon cancer prevention. That's the only study was there. There was a little small literature the NCA actually conducted the study, and the berberin study was there. And also, he started searching about what is this berberin about, and what does this actually do, and things like that. There was a small amount of literature at that time. And then it took some time for him to adjust and, getting along with other colleagues, and there are a lot of MDs, there are a lot of people, English and everything, a lot of things happens there, and the whole environment is different. Okay, scientific environment, morning to evening, they all talk about science and things like that. So when it comes to that environment, it takes some time to adjust and also study, right? There's a lot of cultural shock and a lot of other things, right, when you go to do postdoc in different country, right? And then he took and started doing simple assays like migration assay, like colony formation assay, viability assay, MTT assay, things like that, just to learn. That's what he's going to do when he comes back and he start doing the same assays, right? So that's how I want him to get trained, so that he can come back and comfortably know how to do. Even though you do the migration assay, believe me, there are a lot of things can go wrong. We can tell you what you can, when I see the picture, when I see the analysis, I can tell you multiple things that you may be right or you may be wrong, okay? Things like that. There are a lot to learn in each assays, right? And then he did all these Western blot. Just getting the right Western blot itself, it takes 6 months. So getting the one good Western blot, it takes 6 months to really learn. For some people, they're technically very strong, right? Some very, very weak. So you have to know where you are strong and where you are weak. But he learned because there were a lot of other experts around him, because there are the postdoc, graduate students, they'll be running every day, and anything problem happens that they can correct him, and they're beautiful Western blots, right? So you see the HPNE, HPNE KRAS, they are normal. HPNE is completely normal. See what happens, berberin did not do anything. And also he started with AKT because it is not just only AKT, he actually did a lot of other things. The one AKT where actually antibody was available, we knew he did a lot of literature survey, and he got something. AKT is one of the probably signaling. That's why he selected AKT, and it did work for him. So you can see in the BXPC3 and the PANC1, and also this is the mouse line. In the KPC that I keep talking about, the KRAS and P53 mouse line, that is the mouse line here. So these cancer lines, in these cancer lines, you can see that the phospho-AKT is going down, but not in the normal line, and in the mouse line also it's going down. So it's beautiful, right? So basically he got the target. He got the target, but the target, what you were thinking about the crab, he did not. See what I'm talking about? Even though he got hit, the crab was down completely, but when he started doing, but he did not get it initially. Remember, we published on a paper that AKT can activate crab. Okay, when you inhibit AKT, you also inhibit crab. I also showed you AKT inhibitor also was inhibiting crab. But somehow this compound, it was inhibiting AKT but not the crab. But we don't know. There are a lot of literature we have to do. But for us, at least we wanted him to complete the postdoc period, understand just not the in vitro assay, we want him to take him to in vivo studies. So what he did, he went and did the xenograft assay. So I don't know, he remembers these tumors and the berberins treated tumors and all. So these xenograft assays, at least he can learn some in vivo assays so that he can come back and do something more on those aspects. So berberin basically it inhibited the tumor, and even though the target that we were thinking about, the berberin, so you can go wrong with the chemical. So don't think that okay, you screened and you got it and it's perfect. No, it is not like that. So you have to validate them. That's what I'm trying to tell you. So when you're validating it, you may not hit the target. Okay, you may hit something else, probably that is related to your crab. The AKT is actually, we published a paper in Cancer Research in 2018. I think he was there at that time. So in that paper we showed that AKT is the upstream of crab, and when you inhibit crab, AKT crab is getting inhibited. So that is this story. So let me tell you, this is I was telling Joshi today morning that last 1 year there were 177 berberin and cancer studies. 177 papers came out. Even in the Nature Communication paper, there's a strong, great papers. But just 1 year, I'm not talking about like 18, 19, 20, 21, 22, 23. In each year, probably there are many more papers came out. Just I'm talking about just last 1 year, many papers came out. So when we started, there was only one paper and two papers, maybe a few others, here and there. Okay. When you see something is happening, some of the natural compound and natural medicine, and other people also will be seeing the same thing. Okay. So that means that the winner is who publishes it. Okay. Winner is always who publishes first, right? You had to publish it. Okay. If the data is good, and you should be the winner. No, not others. So you be the winner and start publishing, right? So we did not publish. So I'm just letting you know, because he has to finish the program and came back, and then we did not, because we have other experiments running and we have other targets and other work in the laboratory. So we did not complete. But what his accomplishment, actually he did a lot of other experiments in molecular cancer therapeutics. He published in a co-author, and cancer research communication as a co-author, because he was also involved in other projects. So that when he was involved in other projects, so he was also getting the co-authorship. So it actually help him to get other benefit being a postdoc. So right. So that's one of the example I just wanted to give you, that anything can go wrong, anything can go right. When you are screening compound and when you are selecting something can happen, something not, and also your time factor, right? How long are you going to study this? Okay. So if you would have stayed another 6 months, probably you would have published a great molecular cancer therapeutics paper. Probably you would have figured it out something molecular mechanism. Probably you would have done something genetic mouse model. You would have even in the pioneer in some areas. Probably you would have come back and immediately applied maybe a great grants here. You never know, right? Even though you got other grants, but what I'm saying, the experience will help in a lot of investigators. I'm not saying that you should be in other country to learn these things, okay? You can actually learn the same thing here. There are a lot of great laboratories. There are a lot of pioneers, investigators all around. Look around, there are many of them around, okay? Like we communicate with a lot of investigators. They do fantastic work around, but it's just that you have to find the way to reach them. It's not easy. Okay, you can reach outside, but it is difficult to reach here. But you need to find the way to reach them. So how you reach them and how you contact them and how you interact with them is also key factor. There are great laboratories around here. Okay, I don't want to name the institutions. There are a lot of institutions, even the universities. There are a lot of fantastic laboratories, and pioneers are there. Okay, my pioneers from chemistry is Dr. Rangappa, Dr. Timaya. They were all chemistry pioneers. We learned a lot of things. Dr. Timaya was actually working on he had a 3 project, DBT, DST, and ICMR. All these projects was running in one time. He had a great project. Even the foreigners were coming to his laboratory and working in the laboratory. Okay, and this is in Mysore University. So I'm a biochemist, but I used to go to his laboratory because his lab work was fantastic, because he was actually applying chemistry to biochemistry and biochemical assays, and also biology of it, and tumor and all those things, because he was curious. He's actually thinking internationally, because the people coming from St. Jude's and other universities from Memphis, they were doing experiments in his laboratory, investigators. So I'm just letting you know, it is about you, like finding the investigator where who actually conducting the right experiments and conducting right research, so you can find them. So just giving you an example that it's not about foreign, it's about here also, it's about you. So in vitro validation of this compound, for example, the one example I gave you, berberine, but there are a lot of other compounds that we screened and conducting experiments, that I'm just saying X because we did a lot of other different assays, different studies in different compounds. Every day we were thinking about something, right? One will fail, that doesn't mean that we're going to fail. We're going to come back, and there may be other good compounds that maybe they are active, they may be important, and we keep searching, right? We did get X compound that is actually another compound with a great study, great compound, it's ongoing, and a lot of things are coming out. I just wanted to tell you that it's going to come out, but what I'm saying, we did a lot of in vitro assays. I want to say X so that you know what we did, so that at least experimentally you know what we have conducted, right? We did the dose response studies, we did the evidence-based studies, and transcription regulator, same like what I was explaining to you, the reporter gene assay, and also we did the binding assay, inhibition, Krebs phosphorylation, all these assays we did in vitro, okay. We did again mechanism of action. So we went ahead, like how I showed you the urolithin compound. The same way, we went ahead and did a lot of mechanism of action, just not Krebs, right? How this is, the whole thing is happening. You have to study. It's just not, you just take the compound, okay, fine, it's about your facility, but you need to find a way to understand the whole mechanism, right, as a researcher, scientist, right? And we went ahead and we took this compound and we studied all the cofactors for the Krebs. We did more than we think of, right? And also depends on the person who actually conducting experiment. If they get excited, believe me, the science will change. If they are not excited, then it doesn't matter how much I'm going to tell. It's just not going to work out, right? So you have to get excited. Then only the science will change. Okay? Doesn't matter how much the PI is going to talk to you and tell you. So and also we did a lot of signaling pathway, and also we did a lot of Western blot, phosphorylation, Krebs, different cell types. So we were talking about sensitive and resistant. We also did some studies, sensitive and resistant. Also we did some metastatic study, invasive front, and all those things. And we did chip assay to see what is binding to Krebs and how the Krebs is regulated by other factors, and what this compound is doing, where this compound is going and binding to activate Krebs, and how this is all the interaction, things like that. Okay, just to give you a hint that what all we have done, so that you also know that as an example, you all can do the same thing for your compound and active compound. So what we do in our laboratory, preclinical development to the clinical, I showed you a lot of examples. When it comes to cancer field, why you want to study anything in cancer? Because you want to cure, right? End of the day, you want to cure. Okay, if you want to cure, then you have to do the clinical studies and preclinical and clinical development. That is the idea of cancer biology. That is the idea of cancer research. You have to understand how the cancer grow and progression of the cancer, metastasis. These are all great. This is all biology. That's also very important to that's what we do. We understand the biology, and then we go ahead and do the clinical development. And if you don't understand the biology, it is very difficult to study, right? If I don't know what Krebs is doing and during the progression of the cancer, we don't know where we are targeting, when we should treat this patient, and when we should treat these mice. These kind of things, you have to have the knowledge of the progression and also knowledge of chemical compounds and the screening and understanding the how to study the biology of this. And we also did the pharmacokinetics analysis. So what I'm giving you is a step. When you find something, what do you need to do? Okay. So we already discussed yesterday, other way, the reversal way. But here, we know the target and we are doing the screening and selecting the compound and studying. So we do the same way, toxicity study, pharmacokinetics profiling, and then we evaluate in vivo like PDXs and a genetic model. So we have the patient PDXs and we inject in the mice, and then we study, again use the compound and see whether it works. Translational, like making sure the same thing I'm repeating again from yesterday. The reason I'm telling you, because that study is different, this study is different little bit. If the way that your laboratory or you are thinking, you can actually plan accordingly. Okay, and we finalize a lot of other things, just not biology. Same time parallelly, we select the lead compound and move on, right? So the impact of this next step, you already know. You can actually tell me now what I'm going to do. You know already what I'm going to do next. I'm going to do the chemotherapy, I'm going to do the immunotherapy, I'm going to study further on the tumor microenvironment. You already know. You know my thinking, right? Exactly, it's the same thing. So it's a standard protocol. It's just a standard procedure. Okay. Once you know how this work, believe me, you can do plenty of work. It's the same procedure, same thing. Okay. I'm going to keep repeating the same and again and again. It's the same thing. Okay. I'm going to do the monotherapy and also do the combination therapy. If you want to do the combination therapy, you need to know what are the activators. You saw a lot of activation, right? There may be activation. What I did not mention there, that there are some activators are very important. If they get activated, it is bad for tumor. Okay, you're using some inhibitor and something is activating, even though it is targeting CREB, is bad. So you need to select that inhibitor, block it with the combination therapy. Okay, block it with the combination therapy, and also you do the chemotherapy on the other therapies, and you do the, now next you're going to do genetic mouse model, and you're going to do PDXs, and you're going to do a lot of things, and before you translate your study to the clinic, right? So you all know now what to do. I hope you're going to start working and start thinking it's a standard protocol procedure. Okay, you just do this. We just do the same way. Probably until unless some postdoc and grad students come smarter than us, and even more smarter, and they're all smarter than us, definitely. Okay, each one has a smarter idea and different ideas. Okay, and if someone comes like that and think about something differently than what we are doing, we will definitely change the way that we are thinking experimentally, right? It's sorry, this is not egg, this is a take home message. This is a, now, overall, like it's a compound screening optimization, and can lead to a discovery of, effective targeted therapies. I showed you one of the discoveries, urolithin A, and there may be other discoveries coming. But what I'm saying is that you screen and start, and you discover something that is actually useful to the patient. And also, it actually gives you hope for improved outcome. Either if it is a CREB-driven cancer or AKT-driven cancer, whatever is driven cancer, right? We are saying that CREB is activated in the tumor, but it can be something else, okay? So it actually gives you hope. Oh yeah, I know what, I think I can treat this patient even today or tomorrow, someday in the future. I think my compound, my study will go to clinic and cure someone. Okay, I'm so happy for that. I think that it will happen because I'm actually towards that, right? I think that is the take-home message. I want you to have it. I hope you have taken all this knowledge all these days. Something will be definitely, here and there, something will be helpful to you. And take this message, take this knowledge, and go back and I think start thinking what you can do to cure cancer, right? I think that's important. So I'll take any questions if you have, few minutes. Okay. So now these questions you can actually have questions from even the previous days is also fine. So if you have any doubt on a compound screening, I'm not specifically to the scientific. I want to come up with a general. Like for example, you have something that you don't know what to do. It's okay. So you want to start. Where do I start? Okay, it's okay. And you already started. You got stuck. It's also okay. We can actually try to help you, or maybe give you a hint, or we all try to solve each and others' problems, right? So if you have any questions like that, please let me know. A few minutes. Okay, we can discuss later. So thank you so much. I think we have this discussion forum after your test. So that is the final session of today. So in the discussion, we're going to discuss, like if you have any questions. If not, it's okay, fine. We'll discuss generally. Okay, we can talk about our life. It's also fine. So we will have that session. I think it's probably the tea time. I just don't know whether they are ready. So we can do that too. Yeah, sure.
As you use this array of kinase inhibitors, so they were using 80 kinase inhibitors for covering about 35 kinases. So among these, I have not seen the CREB inhibitor in that.
Because there are no CREB inhibitor, which.
Maybe upward signaling molecules.
So your question was is that there are no CREB inhibitors in the screening. They are all the upstream regulators of CREB. You're right. That is our plan, because we don't have the CREB inhibitor. That's why we went ahead and screened. So if you have the inhibitor already, and why do you want to search for another inhibitor? Rather you study something differently. Or if you want to find a new inhibitor, if you have a CREB inhibitor like already approved, also you can add it to the screen. You can actually, these companies can add it to the plate and give it to you. So our idea, because we don't have we want to find something which actually we can use a clinically approved drug. That's the reason that we screened all the upstream targets and regulators. So we did find a lot of them, but it's just that, remember, as I said, it is not easy to find the one actually is working for you. So again, you have to screen each one. But remember, there is always something come out from the study that you're going to do, right? Or when you start, experimenting these compounds, I think you'll find something new. But important, I think, how you find, and it's about your experience, and also the laboratory people who actually, whether searching literature, understanding the concept, and designing the experiments better, and literature survey, and all these things, all what I was talking about yesterday. It's all about that. It's all about knowledge that you have. Then only your science will change, right? You can do the way, for example, my mentor says, it's okay. But there are I was not. I was like, I was always searching for literature, thinking about what is the new idea, new concept, new design, new thinking. So that we were able to discover more and more. So I think important is that giving the next generation, scientists, like for example, grad students or the postdoc, I hope, and also at the same time the awareness and literature survey every other day, and giving them the time to search literature and collection of literature and discussing with them, having a journal club, and going through somebody else's paper, published paper, and trying to understand, discussing in the laboratory how exactly they conducted these experiments, so that you learn from those experiments and discuss yourself. It's okay, whatever the knowledge that in that room, it's also okay, you can actually discover something. These are all some of the criteria that you can change the way that you conduct those screening and experiments. For example, we take one of the scientists called Channing Durr. He's from North Carolina. So he is my one of the pioneer researcher that I actually study his papers a lot, because he published a fantastic paper in the field, Nature Science, and also fantastic, even the small paper, it's purely scientific papers. And I learned a lot from the scientists. So even I met him and I talked to him in many conferences we go to. He's one of the pioneer, and in our pancreas field, and also in the drug study. He has done a great job in scientific field. So talking to them and learning from them, and also even though he actually went through one of my study when I asked him, okay, can I show you what we did and conducted, and he was so happy to see and give his hope and whatever that we are doing, right? And learning from kind of such scientists is also something that gives you energy. So go to conferences and meet some scientists like that in an Indian scenario, or someone who comes from outside who has experience, and talking to them and taking their time and few minutes and discussing your research and science is also important, right? They all want next generation to be a better scientist than them, right? Or I want next generation to be better scientists than us or me. So happy to help and give whatever the suggestion we can give scientifically, and so that your science will be much better than mine, and you discover more and find better cure than what we have done, and the things like that. So be open-minded, and also same time increase your knowledge and awareness, right? Okay. Let's have tea and then we'll come back. Thanks.